Now we're going to move into the second section, into the stratified epithelium. So I'm very happy that you were able to make it up to here in the video. And remember, we study all the simple epithelial tissue. That when it's flat, it's called squamous. When it has this very organized cuboidal shape or cube shape, is called cuboidal. Here you see the columnar, that it can be ciliated or non-ciliated, and you can have also this false stratification that is called pseudo-stratified. Now we're going to move into a combination of these names, but in this case they will be always stratified. So you will have squamous, yeah, but this will be flat. So here you find all these flat cells here on the top, but and here they will have slightly different shape, but clear there is a stratification. Now you find other layers of the skin that have even, if you see that these cells on the top, they will have layers that protect the skin even more. You will have columnar, and you will have also this one that we will talk, that is called transitional uh, stratified epithelium. That is very interesting because it change, it's very dynamic, it change in shape. Okay, so let's start. In your textbook, they offer us a different classification that maybe is more useful, and they divide it in two types depending on the presence or absence of keratin, this protein that make epithelia very resistant. So the one that don't have it and the one that have it. And they also have another division that is the very interesting transitional epithelium. So let's start. So here, what you can see is an example of an stratified epithelia. So in addition to the first layer, that the single layer that we used to see, here we have 2, 3, 4, etc. We can have as many layers as they have. So for example, in the epithelium of your skin, there could be many more. And of course, the ones that are located in the basal area are the ones that are going through mitosis, and they're generally alive because are the ones that have all the nutrients coming from the vascularized tissue. The more layers that you have, the more likely is that the ones on the top are not alive anymore. For example, with your skin, the top part of the cells of your skin, they have keratin, a lot of keratin, but they are not alive. And this is the next topic that we're going to talk, the type of stratified epithelium. And here we find the two types. First one here, second one here. So they can be divided depending on the presence of keratin or the absence of keratin. And here you see that in the drier areas, yeah, like here, or your skin, yeah, the dry regions, you will find this strong tissue. While in the more wet region, you will find this softer version of a tissue, that is the non-keratinized mucosa, or you can also find it in addition of the lining of your mouth, in the esophagus, and then part of your epiglottis. So to summarize, I told you that the stratified epithelium, remember, contain 2 or more layers, and I will show you example of some of them that have many, many layers of tightly packed cells. Remember that only the basal layer are the ones that receive the nourishment needed to be capable of mitosis, and these layers, yeah, they work as a protective barrier against friction and abrasion, especially the ones that have keratin. And I told you there were 2 subtypes depending on the presence and absence of keratin. And the ones that don't have keratin, they will be in the wet surface, while the ones that have keratin, they will be located in the dry surface. So here I just wanted to show you the importance of the keratin, not only for human, but during evolution. So this protein that is so important to work as a protective barrier is not only found in the skin, as I have told you. And remember, in the skin, It prevents the dehydration at the skin surface, but it's also found in the nails, and in humans it's also found in the hair. And during evolution, it has been used in multiple times in feathers, in animal wool, and also hooves, and also animal horns have a lot of keratin. Okay, so keratin is an important protein that have multiple uses, and is generally related as a shield, as a protective layer. Okay, here you can see a picture of the non-keratinized epithelium. Here you see that the shape is not flattened. However, the one on the top is the one that is actually flattened, that is the squamous epithelium. So remember, it's not that all the stratification are squamous, only the top one. And I already told you the function, that is for abrasion in moist surface. And the locations, I also told you that they could be in most of the lining of the mouth, in the esophagus, and in other regions like the cover surface of your tongue. Now let me show you a picture of a non-keratinized stratified squamous epithelium. Here you can see a diagram of your textbook showing the female reproductive organs, and here they show you that this can be an excellent example of a non-keratinized stratified epithelium. So again, as usual, you have two options. I will give you the answers if you continue, or you can stop the video and think about the answers. So let's start. Here in the first one, it will be this one. I want that you tell me, okay, do you see the nucleus? Kind of. You see a purple, but it's not in the middle. It's like not in the surface. It's very, yeah, is I know that cell is on the surface. So the most likely is that cell is not alive anymore. So that is an example of a non-keratinized dead surface cell. However, when you see the second one, this one, you see the nucleus very clear. Actually, we're pointing at the nucleus. So here you will see the nucleus of a living cell. Very easy. Now let's go to the third one, and the third one is all this giant thing that you can see here. Let me see my pen. It wasn't working, now it's working again. So what you see there is from the top to the bottom, yeah, apical to basal. You see the entire non-keratinized stratified squamous epithelium. And so very clear, I think, so you can see that here is squamous, but here is not squamous as we discussed in the previous slide. And here in the fourth one, pretty easy. That is not epithelial anymore. Yeah, you find other tissue there that look like fibers and cells and all those things that are not as organized. So for sure, there should be connective tissue. So now we're going to talk about the other stratified squamous epithelial, in this case the one that have keratin. And here the first thing that you see is that there is a lot of names. Yeah, and why you have so many names? Because now as you have keratin, the structure of the cells start changing. Yeah, and when we went to the microscope and observed them, we give them different names. Yeah. But in the structure of the epithelial is very similar. Just the presence of the keratin change a little bit the anatomy of the cell. So you will find that we have here on the top of the stratum corneum. Then you will find another one that is the stratum lucidum. Here that is the stratum granulosum. Here the stratum spinosum, and here the stratum basale. And you will find so much information in your textbook about each one of these stratum. Here I'm just going to give you a very summarized version of the problem or what is happening here. So in this basal stratum, what you're going to find is the cells that are going through proliferation. So they have a lot of potential to proliferate, and these keratocytes start maturing, and they start losing their potential to proliferate, and the organs start being programmed to self-destruct. Yeah, so you will start expressing more and more keratin, and the organs start going to deform as a result of and they will become more squamous, and at the end you will have the stratum corneum that the cells are completely degenerated, and you have um, a lot of keratin there, and this is the mature, the last stage of the keratocytes, yeah. And those layers are not dead, are not alive. Sorry, those cells are dead, and they work as a shield, as I have told multiple times, and they can be removed. The second layer, yeah, so I told you about the stratum corneum, very strong stratum. The cells are generally dead, or they are dead, and they have a lot of keratin. The other stratum, the lucidum, so when you went to the microscope and try to observe those ones, you notice that those ones have some granules, but they look lighter than the stratum granulosa. So this is why you call it lucidum from light. Yeah, so they are lighter in color than the stratum granulosum. So in the granulosum, you have all these granules, all these things that are very characteristic. So this is why we call it stratum granulosum. The stratum spinosum, these cells, when they observe in the microscope, they are polyhedral cells that seems to have a structure that look like spines, yeah. And so this is why they call it a stratum spinosum. Okay, so with this we finish these slides. I already told you that the location of this epithelia is generally in the epidermis, where you need a lot of keratin and a lot of protection, especially in your palms of your hand, and in your foot, where it's in contact with the floor. And in the function is protection, again, friction and abrasion, and is again, as I have said multiple times, is found in dry areas. So here I just have again a figure from your textbook, and if you want, you can stop the video and try to guess all these different structures just based on this, and you will do something similar in the lab. So you can stop the video now, or I will give you the answer. So first one, here you see one. And do you see that those cells are squamous? Good. They are on the top. Good. And they don't have a nucleus. Good. So with that information, you clearly see that those cells can be from the stratum corneum. Yeah. And in those case, those cells can be dead. Yeah. In this case, you can say that they are just keratinized cells. Yeah. That keratinized dead cells, dead surface cells. Good. This is enough. Now, here they don't show you all the different stratum. Here they are just showing you, asking you what is that, and here is a cell that is still alive, has a nucleus. Yeah, so textbook is helping you and making your life easier. Now let's go for the third one. That is super easy. Here they are showing you all the epithelial that we just talked. So that one will be, of course, the keratinized stratified squamous epithelium, very long name. And here, as usually, the connective tissue. Now I'm going to summarize all that incredible amount of information. So we talk about the stratified squamous epithelial, two types. One type that have keratin, the other one that don't have keratin. The one that have keratin in land animals is generally found in the skin, yeah, in the epidermis, generally. And in the non-keratinized, just very generally, is found inside of your body, lining, for example, your mouth, the esophagus, and all different structures. If you want to quantify or qualify the effectiveness as a barrier, the one that have keratin are much effective, yeah, than the one that are not keratinized. And if you want to see, okay, which is more likely to have alive cells, yeah, in the outermost layer, the keratinized always is going to have dead cells, like in your skin. The the non-keratinized could sometimes have the live cells, but as I show you here in the previous example, sometimes they could be also dead depending on how many layers they have. And with this, we finish this topic that you can find more information in your textbook. And now we're going to move into the transitional epithelium. Transitional epithelial are very dynamic epithelia. And here in this diagram of your textbook, I want to show you there are these, in this particular case, there are two stages. And I want that you think before you go to sleep, your bladder, yeah, the epithelial there will be relaxed. And if you see, so it will have all these cells that they will be, most of them look like cuboidal, yeah. And when you wake up, yeah, the bladder is full. So what you're going to have is that the cells became have a different shape. Yeah, they will be have the appearance of a pseudo stratified, and they seems to be flattened. And why? Because as there was more liquid in your bladder, now all this space has been reduced, and now it has been replaced by urine. Yeah. So what we have here is an example of a transitional epithelium. And what happened there is that you have in this epithelial a variable shape depending on if the epithelial is relaxed, like I show you in the first one, or distended, what I show you in the second one. And I show you that the locations, it could be in the lines of the urinary bladder, the ureters, or the urethra. So in all these places, it's very important to have this transitional epithelium. Now we're talking about the transitional epithelium, but instead of schematics, your textbook provide this pretty picture. And I forgot to tell you last time that the urethelium is the epithelial that line the urinary tract. Okay, so bladder, urethra, etc. And here I told you that this one has a variable appearance, so this way is called transitional when it's relaxed and when it's filled. And here we're going to talk about the first stage when the cells are partially relaxed. And your textbook show you again the normal structure that they want always to show you. So here in green, you will see this. Oops, I didn't make the correct line, but it's pointing at this. All this structure will be the urethelium. Very easy. And below the urethelium, of course, you will have our friend the connective tissue. On above of that, in this space where there is no cells, you will find the lumen, okay, of the urinary bladder. And if you see, most of the cells in this epithelium will look have a cuboidal appearance, yeah, a stratified cuboidal epithelium. However, if you go and zoom in on the top cells, of course, your textbook, let me put it in yellow. Again, in yellow, this one will look at the nucleus, and here is the nucleus of the transitional cell. The only exception that it doesn't look completely cuboided is the rounded cells found in a relaxed state. Yeah, so those one will look rounder than the other ones, but of course they will change over time when the bladder get filled. So just to recap, I told you two things, that in this epithelium, in the yellow area that you see there, we zoom in, this will be the exception. In those ones, the cells will look, the apical cells will look larger and rounded. However, most of them will look like a stratified cuboidal epithelium. Now let's see what happened when the epithelial goes to a fill stage. Now we're going to focus on the second stage, that is when the bladder is filled. So when the bladder is filled, what is going to happen is that the epithelium is going to have a completely different shape. So if you observe the shapes here, the first thing that I have to tell you is, look, this urethelium now became much smaller, yeah? And as I said before, this epithelium became flatter. And it became flatter. Let me show you an example of that. Here, remember that it was almost like a circular cell, that it was very large. Now it became much more flat cell, more like a squamous cell. Yeah. So here, the appearance of this epithelium, it went from cuboidal to an stratified squamous epithelium. Okay, and of course, here you have the lumen, connective tissue, and generally they show you here is the nucleus of this transitional cell. So let's recap all the information that I told you. I told you that as the bladder fills, the tissue is stretched, and as a result of that, the cells become flatter. The other thing that happened is that it gives an appearance of an stratified squamous epithelium. And what is the function of that? It allows the urinary organs to stretch and maintain the protective lining when holding variable amounts of fluid without rupturing. That is very important. Okay, so with this we finished the transitional epithelium, and now we're going to move into the granular epithelium. Now we're going to show you some pictures of the granular epithelium. So here you have one, and you have another one. And these are two different types of glandular epithelium. And let's talk about what they have in common, and let's talk later about why they have what are the differences. So first, what they both have in common, if you see the yellow cells in both, they are specialized cells that are secreting something. So here you see in blue they are secreting something, so those cells are adapted for secretion. Yeah, so this is what they have in common. What they have different is that if you see this one release things, this one are called endocrine, and this one are called exocrine. Exocrine. Okay, let's later we write all these things. And the endocrines, they will release things in the blood or the interstitial fluid. They don't have a duct. They just secrete things from one region to another one. Here, you clearly see that there is a structure that allows them to secrete things. So they have a duct. And here, no duct. Okay, so while this one will be secreting hormones, for example, in the blood, this one will be secreting different molecules into the skin or the surface the surface of the skin or the lumen of the stomach. Okay, so now let me clean everything, and later we recap what I just said to you. So I told you in the beginning that the definition of a epithelial gland is a single epithelial cell or a group of cells that are specialized to secrete a substance. Okay, and there are two types, and I told you that one of them were the endocrines, that if you remember, they don't have a duct, and they secrete things into the interstitial fluid, or they diffuse it into the blood. So for example, here they show you that they could be hormones that are secreted there. The other type is the exocrine glands, that they do have ducts, and they transport their secretion inside of a tract into the outside of the surface. It could be the lumen of the stomach or into the skin. To talk more about endocrine glands in more detail, your textbook show you that the thyroid gland have many endocrine examples of endocrine glands that you can see here, and they even offer you this additional schematic to make life easier for you. So first thing that we have to tell you is that the thing that you see here is not a duct. They don't have a duct. They are ductless. Yeah, here what they have is a space to store all the hormones, yeah, all the molecules that they are going to secrete. So remember, they are ductless, and here what you see is, of course, what you are seeing is cells with a cuboidal shape that are producing the hormones. In addition to that, here, I don't know if you see here, and you see here, yeah, there's our blood vessel that is a place where they are going to secrete these molecules that are hormones that are chemical messengers. And here they give an example of the thyroid, but there are so many more examples than the thyroid. So hormones can be produced in the, for example, for the sex determination in testes and ovaries. They can be produced also in the pancreas and adrenal gland. There are so many examples. Okay, so let me delete some things and we can summarize everything that we said. So first thing that I told you is that this is a ductless gland and that is made of cuboidal epithelium. And I told you that they secrete things or substance directly into the blood. In this case, those secretions are called hormones that are chemical messengers. And I also told you that there are many examples. So here you can see 6 examples that we will cover later in this course. In addition to that, you can see, I show you here, you have 1, 2, 3, and 4 things that you need to remember. The first thing that I show you here is that this structure, if you remember, and this one, pretty easy, blood vessels. The second thing that I show you is that this, okay, all of that from here to here, it will be the gland. But in particular, this thing and this thing that you can see here, those are the cells that are producing the hormone. Yeah. And finally, the third one is the place. Yeah, the store precursor of the hormone. And finally, the last, the fourth one and last part of this of the endocrine gland is the thyroid follicle. This is a specific of this type of gland. Yeah, that this is a layer, a wall that is covering all the cuboidal cells and grouping them together. Okay, so now let's talk about exocrine glands. The first example of an exocrine gland here, if you remember, I told you they could be unicellular ones, is the goblet cells that we talked before, that they secrete mucus. Here they have all these organelles that we talked in the beginning of the course, that they are in charge of producing large quantities of this mucus. In addition to that, you have multicellular glands that, of course, I told you that they have ducts and tubes, and they use those to secrete these molecules, and they can do it, on the body surface, or they can do it into hollow organs. So let me show you some examples because there are so much variation. So here you can see 2 types of classifications. Yeah, here one type and here an anatomical classification of the glandular epithelium. Here in the left you have the salivary glands, the mammary gland, and the oil glands of your skin, and they have different ways to secrete the substance. So let's start with the first type, that is the salivary gland that have a merocrine secretion. And what you see here is exactly what happened. You see that there are dots that are leaving the cell. Yeah, so what happened here is that you have secretory vesicles that are secreting that. Yeah, there is no loss in the cells. Yeah, so you have the Golgi complex that is working very hard to produce all these molecules, and they are secreted. In the apocrine gland, that is the second case, if you see here in the beginning, you see all the secretion. Yeah, but you are also losing a little bit of the cell. So the cell start pinching off. Yeah, so it goes with the secretion, not all the cell, but a part of the cell. Again, you have the Golgi complex working very hard to produce all these molecules, but there will be some part of the cell that it will be lost. Finally, so the hollow always mean the full, yeah, full. So here you're losing the entire cell. So here the cell will start producing all these all these secretion molecules. In this case, it's an oil gland, so it will be all the sebaceous oil that is produced in the skin. And if you see, it increases, and there will be a point where the cell mature and also dies and become part of the secretory product. Yeah, so it's also secreted there. And but what happened when that cell died? The gland disappeared? No. What happened is that there will be cell division, and so the cells that die, they are repaired and they leave the epithelium. Okay, so I show you three type of secretion that are very different: the merocrine, apocrine, and holocrine. Yeah, and in each one of those, you lose you don't lose anything. The cell doesn't lose anything. You just secrete it through vesicles, or in the other one, you need to produce a new cell because the cell died and it become part of the secretion. Okay, now here you see based on the anatomy, different classifications. So here you have a simple tube that is the one that I start showing you. However, you can have branches, and you can have three branches here. You can have a coil one. You can have here an acinar one. Here an acinar one that it could be also branched, and here you can have a compound tubular, compound acinar, and compound tubuloacinar. So there is a lot of variation, and in your textbook, if you want to know more about this, there is plenty of information. So in addition to all these types that I show you, now I'm going to show you the example here. So here you can have the sweat glands, you have the sebaceous gland, you can, for example, have other ones like the pancreas that can have both. Yeah, it can be endocrine and exocrine. So, for example, the pancreas has exocrine functions that help in the digestion, and an endocrine function that regulate the blood sugar. And something similar can happen with the testes and the ovaries, that they can have both. Now let's move and show you some diagrams and pictures of your textbook, and we can identify the different parts. So I picked this picture of your textbook because it shows something that it looks very similar. It can be confusing with the endocrine glands. Okay, so first of all, I have to tell you here you find the skin. Yeah, and in the skin you have so many glands that we will talk in the next chapter. And here in particular, this is a sweat gland. And we are not going to see the entire the entire gland. You're just going to see a portion of the secretory gland. And you have a sectional view, yeah. So we're going to make a slide cut it like this. Yeah, so you are only going to see the duct. So here, Portland, please, this is not in number one. Yeah, number one here, you are not going to have a storage place. This is the lumen of the duct. This is, where all the material is going to go outside. That is the place, the tube that where all the secretion goes outside. The second place here you have in both, you see that is the nucleus that is constantly active, producing all the proteins that are going to be secreted to the sweat glands. And in the third, of course, you will have in both the basement membrane that is very clear in this case. Finally, I just wanted to make a point here that in this case, you are not going to find this simple cuboidal epithelium. You are going to find here two layers. Okay, and with this we finish. There's much more we can cover that, and we will keep covering these glands in more detail in different chapters, but I think this is more than enough material to cover for this class. So we covered so much topic. So in the beginning, we talk about what is a tissue, and I told you that tissues can be defined in different ways, but the idea is that they have group of cells that they can be different, but they have a similar origin. They can come from a similar germ layer, and they all work together to perform one function. Yeah, so and I told you about 4 tissue types: epithelial, connective, muscular, and nervous tissue. In addition to that, I told you that there are very important protein complex that allow the cells to attach to each other. They work like in some degree like cement, that they allow one cell attached to the other one, and attached not only to this one, but also attached to the basement membrane, so here. Okay, and in addition to that, so we finish what is an epithelium, that there are 4 types of tissues, and that we talk about cell junction. Then I move into the epithelial tissue, that they are these group of tissue that have a lot of variation. Yeah, and the main function is covering or lining structures. I told you that there are different ways to classify epithelium. So for example, you can classify it based if they have single layer or they have multiple layers. And also you can classify it based on the shape of the cells. And we cover both simple epithelials and stratified epithelial, and later at the end we cover some glandular epithelium. Okay, later we will move into the connective tissue. I hope that you enjoy this section of the class, and if you have any questions, just let me know.